Hey everyone, this is my lesson 9 group video for group problems. Just to go over the problems here. Okay, so for question 1, ask us to diagram intermediate stage of DNA and asks us to identify the ORC. So the ORC is right here at the top where the 3' and 5' start together. And this is the origin of the chromosomal replication, which is where it starts. And then the DNA primase. DNA primase is right after the DNA helicase, you can see here. DNA primase synthesizes with DNA polymerase 3 alpha to create the leading and lagging strands, and also synthesizes the RNA primer. Right here you can see the RNA primer coming right out of it. And then the DNA helicase is right here, this structure right here. The DNA helicase is recruited to separate and continues to separate the two strands of DNA. Here you can see how they separate after they're exposed to the DNA helicase. And the RNA primers, you can see, are coming off of the lagging strand right here, the bottom strand here. So the lagging strand is between the third Okazaki fragment. So there's one right here, the beginning of the Okazaki fragment, and then at the end, Okazaki fragment, linking it to the second and first Okazaki fragments that have already been connected together here. And then the RNA primer on the leading strand is on this top strand right here, right after the DNA polymerase 3 alpha. So it's coming right out of there, it's connecting it to the replicated DNA strand. And the two leading strands are, my first one up here is going to be the blue DNA strand. And that's going to be the strand that goes from 3' to 5'. And then my second leading strand is going to be the black one here on the bottom. And that's the strand that goes from 3' to 5'. And then, let's see, 5', yeah, 3' to 5', yeah. And then the DNA polymerase 1 is going to be this structure here and right here. There's two of them that I've drawn here for both replicated strands of DNA. And DNA polymerase 1 removes the RNA primer from this strand and then links it to the rest of the DNA, or links it to the rest of the DNA strand. So it takes that RNA primase out of there so that they can, those two strands can link together. And then the DNA polymerase 3 alpha subunit is going to be the subunit here, and this synthesizes the leading strand and the lagging strand. So you can see that after they're exposed to the DNA polymerase 3 alpha subunit, that we now have leading strand, lagging strand, and on the bottom, same thing. We have the leading strand and the lagging strand. And for the Okazaki fragments, I kind of already went over that, but right here we can see the Okazaki fragment. The third Okazaki fragment is between these two RNA primers, and then the second and first Okazaki fragments have already been connected together, and they're on this last part of the DNA segment right here. And in question 2, fill in the blanks. So this protein complex synthesizes primers in eukaryotes, and this is going to be the DNA polymerase alpha. And then this protein promotes the separation of the AT-rich sequences within ORC. This is going to be the DNA A. And epsilon is a subunit of DNA polymerase III that contains proofreading activity. And for D, the DNA polymerase epsilon is the protein that synthesizes the leading strand in the eukaryotes. For E, the TUS, or termination utilization substances, this is the protein that displaces the DNA to polymerase III from the chromosome to terminate the replication process. And for F, DNA polymerase I is the protein that has the 5' to 3' polymerase and the 5' to 3' exonuclease activity. For G, the topoisomerase type 2, which is also known as DNA gyrase, is the protein that introduces the negative supercoils into the DNA ahead of the replication fork. And for H, the origin recognition complex, or the ORC, is the protein that binds directly to the ARS elements. And for I, the single-stranded DNA binding proteins, or the SSBPS, is the protein that prevents DNA strands from annealing during replication. And then DNA ligase is the enzyme that seals the Okazaki fragments into a continuous strand of DNA. And for question 3, we are identifying enzymes involved in DNA replication. So for question A, the 5 enzymes that consume ATP during DNA replication are DNA helicase, DNA gyrase, DNA primase, DNA ligase, and DNA polymerase. And for question B, I only got 3 of the 5 on this one. I wasn't really able to do any of my own research past what the notes said for the 5 enzymes that consume DATP during DNA replication. But so far I have DNA polymerase I, DNA polymerase III, and then DNA ligase. And for question C, the 5 enzymes that have the 3' to 5' exonuclease activity, that's going to be the DNA polymerase III alpha, DNA polymerase III beta, gamma delta, chi, psi, and then epsilon and tau theta. And then for D, the enzyme that is involved in replication that uses DNA as a template to synthesize RNA is going to be that DNA primase. And then the enzyme that uses RNA as a template to synthesize DNA is telomerase. And for question 4, you can see I drew out the template up here. And then for question A, since we're using the A, C, G, and T nucleotides, I just wrote out the complementary DNA strand. So I started with 3', ended with 5', and just made the complementary strand. A is paired with T is paired with A, G is paired with C, G is paired with C. And then same thing for B, we're using the A, C, G, and T nucleotides. So again, I just started with that 3', went to 5', and paired them together to make that DNA complementary strand. And for C, we're using the A, C, G, and U nucleotides. So A is going to pair with U, T is going to pair with A, G is going to pair with C, and C is going to pair with G. So I just made that complementary strand here. 3' to 5', and made that complementary RNA strand. And then the nucleotides used in D are going to be the A, C, G, and T. And so again, I started with the 3' to 5', and pair the A to T, T to A, C to G, and G to C, and make that complementary DNA strand. Then for 5, we're referring to proteins used in DNA replication. And so for A, the ones that are found in prokaryotes are going to be the DNA gyrase, the TUS, the DNA polymerase epsilon, DNA primase, and the DNA polymerase 3 alpha subunit. And then the proteins that are found in the mouse musculus, which are basically going to be the proteins that are found in eukaryotes, are going to be telomerase, PCNA, CDC6, MCM helicase, and DNA polymerase alpha. And for C, the proteins that are involved in chromosome compaction, I only got one of those. I got that it was the telomerase. And for D, the proteins involved in the initiation stage of DNA replication are CDC6, DNA-A, and MCM helicase. And then the proteins involved in the termination stage of DNA replication of the listed ones is just going to be the TUS. Cool.